friend Yasser. Dr. Yasser Iturria Medina is a neuroscientist, but developing a lot of methodology. He is assistant professor in the Department of Neurology and Neurosurgery at McGill, and he received his PhD in neuroimaging and neuroinformatics at the National Center for Scientific Research and Havana University of Medical Science in 2013. He came to the MNI as a postdoc in 2013 before being appointed as an assistant professor in 2018. So Dr. Iturria Medina lab is the name of the lab is Neuroinformatics for Personalized Medicine. The lab focuses on defining and implementing multi-scale and multifactorial brain models for understanding neurological disorder and identifying effective personalized intervention. So the lab combined between molecular, imaging, and cognitive data using integrated mathematical modeling to create both individuals and also population-based mechanistic brain models. So we talk a lot about brain modeling all the weeks, and then so you are going to see something like very close to the application, moving from how we got the data and what are the focus in the clinic for cognitive science and how we bring those methodology to answer those questions at the individual level or at the population level. This is really a great research integration of the best of what we do in model. And so Dr. Iturria Medina research is focusing on creating and validating of integrated molecular neuroimaging and computational tools for understanding complex causal interaction among aging, neurodegeneration, and different therapeutic conditions. So for all of this, so thank you very much, Yasser, for accepting our invitation, and the floor is yours.
Thank you. Hi, everyone. It's my pleasure to be here today, and I want to thank the organizing committee and in particular Habit for the invitation, and also the public and the people that is attending for being present. Today, or in the first talk, I will talk about the importance of using data-driven modeling for better understanding neurodegenerative disorders and for identifying and also identifying effective disease-modifying interventions. The first talk would be, I would say, more oriented towards explaining our motivations or the way or the path that we follow it to arrive to the research that we are doing today. And the second talk will be specifically focused on in that research. I like usually to start with this slide because it reflects very well the critical situation in which we are. And a meta-analysis study published in 2015 reported that the top 10 sold drugs in US were only helping significantly from a 4 to a 25 of all the patients that are taking them. And these are the top 10 drugs. So the most prescribed, the more used for the patients are only improving significantly the clinical conditions of a small fraction of all the patients taking them. Just to mention 3 examples, the most popular drug for schizophrenia is only helping considerably at 20% of all the patients that take it. The most popular drug for depression only helps about a 12%. And for multiple sclerosis, only a 7%, which is really, really low and significant. I would say very surprising for what we expect at this point from medicine, right? And this situation is not only particular to schizophrenia, depression, this kind of neuropsychiatric disorder. It's also like for Alzheimer's, for Parkinson's, for neurodegenerative disorder, it's even worse. We barely have treatments that work. We don't have. We're starting to see 1 or 2 interventions that seems to be providing some benefits. And even there are a lot of discussion associated with that. So they shows or they support or they highlight the critical need that we have to understand better neurodegenerative or neurological disorders in general from a multifactorial causal point of view. And the second urgent need that we have is to pass from generalized medicine to personalized medicine, from medicine that assumes that all the patients with the same diagnosis need the same treatment, to a medicine that considers the individual predisposition to reply to a treatment or not. One of the major challenges that we have when we are studying progressive disorders is that they can take decades to develop. So usually, and I just want to start with the Alzheimer case or the Alzheimer example. So if we consider, let's say, a person that developed or is diagnosed with Alzheimer by the time that is 70 years old or 80 years old, and normally what we have done in research is that we start studying and comparing subjects with Alzheimer versus those without Alzheimer, right? And there is a problem there, is that by the time that they are diagnosed with Alzheimer, they probably have been 20, 30 years accumulating neuropathological effects in the brain. And there are so many changes at that time that we don't really can identify the early disease triggering effect. So a person that, say, is diagnosed with Alzheimer at 80 already have an brain abnormality, so the brain was not working properly since the person was in the 50s or in the 60s, right? That's a major challenge, how we identify or how we detect early who is into the path to develop Alzheimer and what is that variant of Alzheimer is developing that person. But in addition to that, there are genetic factors whose influence start accumulating since the person is born. So in addition, we start individually, depending on our conditions, influence, etc., we start constructing our cognitive resilience since very early age. And there are also lifestyle and comorbidity conditions that will determine or predetermine how well or not we resist or how much resilience we have to the neuropathological factors that conduce to these diseases. So when we talk about resilience, in this context, resilience means to, and you might or not be familiarized with the content, it's like how cognitively affected we are for a given level of neuropathological alterations in the brain. So there are studies that in the 90s there were some studies showing that 92 early 2000 that some patients with or some participants with the brain full of misfolded proteins or toxic proteins and a lot of structural damage, they were cognitively normal. And others with just a part of that or a similar level of neuropathological severity have a lot of cognitive alteration. So this resilience have been studied a lot in the last years as alternative ways to find interventions that could improve the individual resilience to the neuropathological alterations. We will discuss that mostly in the second presentation. This complex situation is not only typical for Alzheimer, which is the most prevalent neurodegenerative disorder, explaining or covering almost 62% of all dementias, but also to vascular dementia, Parkinson's dementia, frontotemporal dementia, Lewy body dementia, so many other different disorders or diseases that are equally or similarly complex to Alzheimer's disease. Of course, once that we know that these disorders are super complex, that they take decades to develop, when do we start? Let's say if we want to study them, if we want to help to identify or to understand better the disorder and to identify better treatments, where can we start? So there are so many possible directions, so many different ways to study a given disorder. So what we did in our specific case, that we went back to the classic hypothesis of Alzheimer, and we also study Parkinson, ALS, etc. But for now, I will focus on the Alzheimer case. One of the most interesting hypotheses of Alzheimer is the disconnection hypothesis, which proposes that loss of synaptic transmission leads to the reduced activity of targeted neurons in cortical areas, or that brain dysfunction and cognitive impairment, clinical deterioration, might be better explained by a disturbance on the connections between brain areas rather than by specific alterations on specific brain regions. There are other hypotheses we will pass to them later in the presentation, like the prion line, metabolic hypothesis, vascular, neural activity, dependent degeneration hypothesis, and all of them make sense to some extent. There are evidence in favor and against each of them, and all of them for me are also interconnected, and we will talk later about ways to explore them at the same time. However, how to study these diseases in terms of the disconnection hypothesis? As you know, complex brain networks are devoted to the representation analysis of anatomical, functional, morphometric, metabolic brain connections. And they can be devoted to the analysis of any physical network in the brain and also indirect networks, depending on how the network is defined. In terms of brain networks, we have about 2-3 decades now studying anatomical connections identified via in postmortem data, identified with track tracing studies. Functional connections identified with electrophysiological techniques like EEG, MEG, fMRI, and others. Morphological connections, which reflect the covariance between different brain areas across a population, and anatomical connections identified with fiber tertography techniques. I believe you already have some presentations describing fiber tertography techniques. In general, all brain networks share, independently of how they are identified, they share attributes that are not typical of random graph or regular lattices. Like they have high clustering and modularity, hierarchical organization, high efficiency and optimization, central regions, lower structural motifs that are explained on the functional side, that are explained by anatomical conditions or anatomical architecture. They have been used to study neurodegenerative disorders, like in this study on this report from Lowe et al. From 2010, in which they identified a specific brain connection different between the controls of the non-cognitively impaired participants and the subjects with Alzheimer's. And they also identified which specific network attributes, like non-efficiency, for example, reduced predominantly in frontal brain regions of the patient with Alzheimer's. Interesting, they also found that the alterations in the networks in network metrics associated with efficiency, clustering, somehow summarizing the ability to transmit information across the network for each patient, correlated with verbal learning tests and mini-mental state examination, which, as you might know, are a specific tests used to characterize clinically the patient with Alzheimer's disease. This suggests or supports that the deterioration of the network might be associated to the clinical decline that we normally see in these patients. But this kind of analysis is more focused on group comparisons or correlation for a given population. We can also focus on making predictions with the information that is in the brain network, right? So, like in this study, in which we focus on the mice in the shivering mice, which is a relevant animal model to study multiple sclerosis. And what we did was we have very high very high resolution data, like 80 microns from diffusion MRI imaging, and then we use a specific different fiber tertography techniques to sorry, the pointing is not working well. To track or identify or estimate connections between the different brain areas and obtain the brain networks associated with GIS, with the mice, with the for the controls and for the diseased mice. Then, based on the individual brain networks, we estimated graph theoretical measures that allow us to represent the mice in multi-dimensional space, in which were it was very clear that they will be positioning in different areas of those space. And then we use a specific machine learning techniques to predict or to assign individual probabilities to be on the control group or on the disease group. And that worked perfectly. The accuracy was 100% in some cases, etc. And this was just classifying the subjects in terms of groups of belonging to one group and other, but there are a lot of studies after that point. A lot of studies focuses on making predictions not only of the disease state, of disease diagnosis, but also of the disease severity, so that we can also use individual brain network data to predict how close is a person to develop the disease, if a person will convert or not in a few years to a given disease. We published a review paper making or clarifying the distinctions between network analysis for just group comparisons, so making analysis of difference between different disease states, and the distinction with making a prediction of disease advance. And since then, as I mentioned, a lot of other studies have extended this and have incorporated not only network information from one data modality, but from multiple modalities. However, until here, what I have shown are ways to use the network information with the idea of identifying what changes across the disease spectrum in terms of connections only. But it might be the case in which, or it is the case that even when connections could be intact in some cases, let's say one person may have perfect connections in the brain, or connections that are normal compared with a person that is not developing a disease, and still those connections can be contributed to the disease progress, right, or the disease progression. And why that happens? So essentially, well, there is another hypothesis of neurodegeneration. That is the neurodegeneration hypothesis, which in the context of Alzheimer's is strongly related to the amyloid cascade hypothesis. In Parkinson, there is a alpha-synuclein hypothesis, which proposed that in neurodegenerative disorders, there are myophotoproteins that are toxic that start accumulating in specific brain regions, and from them they propagate via the anatomical and the vascular connections to other brain regions, spreading the disease factors. So essentially, in this case, the connections are facilitating the propagation of toxic factors from initial regions to other regions that get altered after, and then from then the toxic factor goes to other regions, and then at the end, the patterns of atrophy that we see, or the patterns of brain alteration that we see, that essentially depends on which was the initial region that started accumulating those toxic proteins and the connections of those regions, then that pattern is what determines the clinical alterations that we see later, right? So if the epicenter, let's say, of one disease is one specific brain area, due to the anatomical connection of the area, is very, I would say, or goes most directly to other specific brain areas, the clinical alterations that we see normally for one individual will depend on the connectional pattern of the initial region that started developing the disease. Normally, we tend to think in this process as something that takes decades, or like, as I mentioned before, the accumulation of neurotoxic proteins takes from 20 to 30 years until the point that we start seeing clinical alterations. However, the intrabrain propagation of myophotoproteins is very fast, can be very fast. In only 30 minutes, myophotoproteins can travel almost the whole brain, and that has been verified in animal experiments. So this was a hypothesis proposed that we say, well, it have some It was proposed decades ago, but I think that it was highlighted again or taken back about 2000, at the early 2000s. And but there was no evidence. There was evidence from the animal study, and the evidence on the human brain was coming from post-mortem study, in where we could see that different areas that were connected were also the most altered area for at the time of neuropathological evaluation. However, in 2009, a study in Europe shows evidence supporting the neurodegeneration hypothesis. Seeley and Koresh collected data for multiple disorders, including Alzheimer's, behavioral frontotemporal dementia, aphasia. Sorry, I don't remember the name of the other. But then what they did was to determine or estimate atrophy, the average atrophy, the typical atrophy for each of these disorders, and then analyze if the brain connectivity could explain those atrophy patterns. And then what they observed was that the atrophy of each specific disorder was overlapping a specific anatomical and functional connectivity patterns in the healthy brain. So essentially, if we decompose the brain connectivity of the healthy brain, we will find a specific mode. So let's say principal components of connectivity patterns, right? Each component was associated.
Oh,
Yes.
I think you lost your Wi-Fi connection.
Yes, right. Yes.
So can you go out from this?
It's working again.
Oh, it came back?
Yeah.
Okay, so you need to connect to Zoom again, to the same link.
Zoom is
It's
starting.
okay. It's not your fault if the Wi-Fi doesn't work. Oh, it's installing. Yeah. Okay, we're back. So can you mute? Okay, share the screen. You can share on, for example, the whole screen. Yeah, just click on it. And go to presentation. Diagram. You can minimize this. Oh, yeah.
Oh,
You can minimize this if you want. Okay.
Apologies, we lost the Wi-Fi connection and then Zoom decided to update in that specific moment. So as I was mentioning, if we decompose the typical patterns of brain connections, like in terms of anatomical and functional connections, we will see that those patterns align very well with the atrophy that we can find in minor or very prevalent neurodegenerative disorders. And that's very interesting, and that supports the idea that depending on where the toxic proteins start accumulating, then it will just, if it's closer to one given pattern of connection, then it will follow those major connections and will affect the specific brain areas, which then will project or will be projecting or affecting a specific set of cognitive and clinical abilities and domains. Seeley and colleagues also show that if we estimate network properties associated, or let's say the distance to a specific hypothesized epicenters of the disease for each specific disorder, then we will see that there is a relatively strong correlation between the atrophy of all the brain areas and the distance to the connectional distance to the epicenter. So the farther is an area from in terms of connections to the epicenter of the disease, the less affected it will be. On contrary, the closer or the most connected is an area from the epicenter, the more affected it will be. That was a very elegant, I would say, data supporting the neurodegeneration hypothesis, but it still was observational, right? So we aimed by 2012 to use mathematical modeling to describe and characterize better this process. I'm thinking on that, and one of the first modeling of neurodegeneration that we did from the mathematical point of view was to use epidemic spreading modeling to, according to which the change in the probability of observing misfolded proteins or toxic proteins in a given brain region will be a function of the probability of receiving these misfolded proteins from the other connected brain regions to this one. And to know which brain regions are connected or not, then we can use probabilistic networks with information from 5 electrography techniques, which will be just providing index of or scores supporting the connection between or not between every pair of regions. Then the change in probability of observing misfolded proteins in one region will be, as I mentioned, proportional to the probability of receiving from the OS minus minus the probability of clearing those misfolded proteins in that specific brain region, which, as you normally happens by the immune system. So the misfolded proteins accumulate in one region, they can be cleaned by the vascular system, by the immune system, and specific defense mechanisms of the brain. I will mention some of them after. Plus less stochastic events and standard inputs, like if the person is taking medical drugs, which kind of diet is following the person, if the person is doing physical exercise or not. Mathematically, this general model can be translated to a set of differential equations that depends on two main parameters. One of them is the production level of the misfolded proteins, so the production rate of the misfolded proteins at each specific brain region, and the clearance rate of the misfolded proteins at each specific brain region. In addition, they depend, as I mentioned, of the connectivity matrix that contains information about how the different brain areas interact or connect anatomically in this case. And it can also depend on a delay time that we assume it will proportional to the distance, geodesic distance in the brain between the regions. But then when we use that information about how fast the misfolded proteins propagate between any pair of regions, it was so fast that this term, the delays were close to zero, and we ignored them from there. So then we used real in vivo amyloid deposition data quantified with PET imaging to quantify at the individual level the level of deposition of amyloid in different brain areas. And in combination, we did that for about 700 participants from the Alzheimer's disease neuroimaging initiative, which is a major study collecting all sorts of many different sources of data for patients in the spectrum of Alzheimer's disease and also for healthy controls. So the amyloid PET is a way of quantifying the deposition of these toxic proteins in the brain, and until I would say 2010, 2000, entered the first decade of 2000, it was not possible to quantify amyloid deposition in the in vivo brain. And now, I was about 10 years ago, also there are new imaging techniques to quantify tau deposition too, which is the other major player in Alzheimer's disease. So once we quantify the level of amyloid deposition in the brain for all the participants, then we use a brain network information in combination with our epidemic spreading modeling to simulate about 50 years of life for each participant. And we move from region to region, simulating how the propagation will be from that region for a given set of model parameters. And we did this in an iterative way that allows us to identify the production and clearing rates and noise level for each data, for each individual data, and also which epicenter region was the optimum region to explain the real data that we were obtaining from the imaging side. After we did that, we finished with individual values for production rate of amyloid in the brain, cleaning rate of amyloid, level associated with the individual data, and also what time or what we call like onset time of disease spreading each subject was having. So that time was, in other words, was like for how long the misfolded proteins have been spreading for each participant, right? Then we observed that the model was able to explain relatively well with high accuracy the stereotypical patterns of amyloid deposition across the different clinical stages. So what we have here in the first graph, we can see the association between the observed level of amyloid and the estimated level of amyloid with the model starting from 0. Sorry, the pointer is not helping too much, but essentially we observe a high correspondence between the real data and the reproduced data with the model. And this also shows that there was an increase in amyloid deposition in the brain with the disease advance. That part is not surprising. We know that from literature. However, the model was able to capture, starting from almost 0 value in given epicenter regions, the typical patterns of amyloid deposition for the clinical cases. We also identify the posterior cingulate cortex as the optimum or the most likely epicenter for Alzheimer's disease. We know from literature that is an early region involved from early in the disease process. However, there are potentially it's not the real epicenter of the disease. What we know, that the region that we know that accumulates earlier amyloid and tau and the entorhinal and the hippocampus in the brain. However, they connections, they are kind of peripheral regions compared with the posterior cingulate cortex. So we believe that the model finished identifying the posterior cingulate cortex because it's a myopropagator, while the entorhinal and hippocampus region were like peripheral regions connected to this propagator. And due to limitations on the connectivity information, usually the connectivity to the entorhinal region is not so well estimated. The model kind of identified the next region that is strongly connected to hippocampus and entorhinal region, which was the cingulate cortex. Then we were interested in to analyze how the model biophysical model parameters or entities that we estimated associated with the clinical advance of the disease, right? So then we did a specific statistical analysis to see which parameters were the most associated with the clinical stage of the subjects. And we observed that amyloid clearance rate was the most significantly associated parameter to the advance of the disease. And what we have here, a different graph showing the relationship between the clinical stage and the specific model parameters. And as we can see, the clearing rates change strongly from the healthy controls to the subject with Alzheimer's, suggesting that the accumulation might be happening due to a lack of sufficient cleaning in the brain of the subjects that are developing the disease. We then calculated the ratio between production and clearance, and we observed that the persons that develop Alzheimer's disease have a significantly higher rate of production versus cleaning compared with those that stayed in the healthy state. We suggest that a deficiency on cleaning these misfolded proteins, which could be due to the immune system, failures on the immune system, failures on the vascular system, and the component of the vascular system that helps to clean the garbage, what we call the garbage of the brain, might be contributing to developing these disorders. And this is an interesting case of how we can use mathematical modeling that combines network information, regional information with molecular PET imaging to study biological or to infer biological processes of a complex disorder like Alzheimer's disease. We then went back to the literature, and in 2 years before that we published that study, there was another study in Science that reported that while for many years or decades, it was considered that the persons that develop Alzheimer's have an overproduction of amyloid in the brain. In fact, what the data was suggesting, or the data was showing, when they injected an isotope in the blood and quantified comparing the level of production versus clearance, what the data showed was that there was a deficiency on clearance too, and not an overproduction, what was contributing to the accumulation of the misfolded proteins. And this was based on other kind of analysis, which all imaging and mathematical modeling were aligned with. In addition, we found that failing in clearance was associated with the number of ApoE4 allele copies that the patients had. As you might know, the ApoE4 is the myogenetic risk to develop Alzheimer's disease. So what we found out was that the person that have more alleles of this gene, they have, the more alleles they have, the less clearing capacity their brain have to clean themselves from those misfolded and probably from other components that will accumulate in the brain. And these ApoE4 genes were also significantly associated with how long the person will be, how early the person started propagating and accumulating these misfolded proteins. The more allele copies, the earlier the person started having these misfolded proteins accumulation and propagation in the brain. We also try to see how these epidemic spreading processes that we just characterized for the brain, for the human brain, could align with effects of similar epidemic spreading processes that occur in society. And what we have here on the figure on your left is the relationship between the effective anatomical distance to the epicenter region that we found. And in this case, effective anatomical distance is just a number associated that reflects the number of connections, or that's inversely proportional to the number of connections to the epicenter, right? So the more connections, the farther is the region in terms of connection to the epicenter, the more time the misfolded proteins will take to arrive to that region. We observed that, and we observed that there is a strong linear relationship between the effective anatomical distance to the epicenter and the time that the misfolded proteins take to arrive to the region in the brain. And then we looked to the literature if similar effects have been reported, and we found that a very similar effect was observed for the spreading of the influenza that occurred in 2009. You might remember that case when there was an influenza that spread across the whole world. And fortunately, data associated with that epidemic was saved and analyzed. And this study reported that the distance in terms of number of flights to the epicenter country, that I think was Mexico, reflected very well how the time that the influenza will take to arrive to those countries. And then that was a very elegant illustration of the role of the network on how information, which could be good or bad information, like in this case for the influenza or for the misfolded proteins, propagates through the networks. And of course, it's a direct measure. We saw also that with COVID, right? The more connected we were with people with COVID, could have negative effects into us. So it nicely showing how the good and dark and bad side of the networks, right? So the networks can be very useful, or are critical for the spreading of the nervous information in the brain, but they also serve as a conduit to the spreading of the negative factors or the diseases that we might develop. However, until here, I only have talked in terms of brain connections and how they can help us to study the first, how the alterations associated with disease advance. Second, how even when they are altered or not, they allow the spreading of negative or toxic agents across the brain. However, we need to consider that any disease state or any brain state and also disease state is more than just what happens at the level of all macroscopic connections, right? So any brain state is the result of the interaction between proteins, synapses, neurons, neuronal circuits, and networks, and what happens at the networks level. Right until now, we have only been focused on what happens at the network level, and specifically, we have been focused on anatomical networks. How can we start incorporating all the layers of biological information? So, just a second. Multi-layer networks is another, I would say, mathematical framework that allows to consider not only connectional information corresponding to a given type of biological or even transportation way, but also they allows to aggregate this information and to study these multidimensional networks in a way that we still can extract or learn from how the information propagates across those networks. Here we have, for example, a multi-layer network showing how could be the transportation, how is the transportation in a city, and illustrated how the persons can go from one side to the other using different vias of transportation, like could be the train, the bus, the airplane, the taxi, the Uber, etc. So in similar way, we can use multi-layer networks to integrate different type of brain networks information. Like in this study from Battistone and colleagues from 2007, they used DTI connectivity or diffusion of fiber tractography information to define anatomical brain networks and fMRI data to define functional brain networks. And then they establish individual brain networks, individual brain multi-layer networks that have both anatomical and functional information. Then they show how we can estimate almost all the same network metrics that are defined for unilayer brain networks, like the efficiency, clustering, path length, global efficiency, small world index, etc., to these multi-layer networks. However, we can go beyond that. So until now, we have been talking of brain networks in terms only of synapses. So which can be, we have the structural anatomical network. What we do is that we have a representation of the synapses connecting one region versus the other ones. And the functional one is essentially the information that propagates across those synapses. But other studies have proposed to use multi-layer networks to not only consider synaptic brain connectivity, but also chemical brain connectivity between the different areas. And the C. elegans case is a very elegant case because, as you might know, the C. elegans is a small creature which all neurons connections have been very well mapped, and that's why it's so much used, because we know how the brain connects, not only at the synaptic level, but also chemically, electrically, and then it allows to apply new developing techniques or already developed techniques to see or to study the properties or to see how do they apply to the brain, to a complex network. In this case, this offer combined information coming from synaptic, gut junction, monoamine, and neuropeptides to see how the connections in the C. elegans and the regional patterns of connections associated with different types of neuron-neuron connections. And they illustrate very well how the synaptic connections, why they are the connections in which we focus the most, on the neuroscience side, and not the only important way facilitating the interaction between the different brain between the different functional components of the living of a living creature. We also can use multi-layer networks to define or to study connections across multi-omics molecular layers. Like, for example, we can see how genes, or we can map how genes interact with proteins and metabolites, and we can study these connections across the different layers of molecular entities, and this can allow us to see how they cluster together, or how genes, a specific set of genes, cluster on target specific proteins and metabolites to allow the functioning of specific molecular processes. And I will present some specific applications using multi-layer networks with molecular omics in the second presentation. How I will show how they can be used to study Alzheimer's disease and other neurodegenerative disorders more into practice. In our case, we wanted to use multi-layer network to combine information coming from anatomical and vascular components of the brain in combination with dynamic system analysis. So if you remember before, the epidemic spreading model that I presented before, considering how the change in a given brain region, change in accumulation of toxic proteins in a given brain region, were associated to the probability of receiving these proteins from other connected regions, connected via the only via the anatomical network. We extended that model now to a multivariate or multifactorial set of components. And we assuming that the change that we observe in a given biological factor at a given brain region, which could be changing accumulation of photoproteins, let's say increasing amyloid deposition or increasing tau deposition, but also could be any other change, could be atrophy increase or hyperactivity in that region, or vascular dysregulation in that region, will be proportional to the local multifactorial interactions in that region. So let's say how we are talking about amyloid deposition in the brain, how amyloid deposition can be impacted by changing the vascular flow of the brain, or by changing neuronal activity, or changing atrophy or the structure of the cells. So essentially, this component here will be associated with all the direct effects, not indirect, direct effects or direct interactions between different layers of biological information, right, or biological entities. So at the end, this here is a term that should be capturing the causality, the cause effects of interaction between different elements within the brain, plus incoming effects from other brain regions, right, which can propagate through anatomical connections, vascular connections, any physical system on the brain. Plus external inputs, which, similar to before, could be if the person is taking drugs, if the person is doing physical exercise, which kind of diet, etc. Then mathematically, we can translate these general models to a differential equation. This is just a simple case, a linear case, but on the left, what we have is the ds, in the term ds dt will be essentially representing how the brain is a stage, a space change in time, and s here can be is a vector with numerical values associated with the level of vascular flow, toxic proteins, atrophy in the brain across different brain areas, right? So imagine that what we can do is to say change in time across all these properties are a function of the local interactions captured by the matrix A between the different biological factors, and also how the information propagates, plus external inputs captured in this case by U, right? And then we could have a second equation in which we project all the properties of the brain and the change in time to cognition, right? Because at the end, that's what we are interested on, right? To see how the person behave, or to study how the person behave, or how decline cognitively as a function of the brain properties. I will not enter too much in mathematical details here. This was just a linear equation, relatively easy for you to follow. But in general, what we're using is dynamic system analysis in combination with multi-layer networks. And as you know, the dynamic system analysis and control theory are used every day in many engineering applications that we use, like in civil engineering, aerospace, mechanical engineering, electrical engineering. In practice, what we do is that we consider two kinds of connectional information, vascular connectors between the different regions. So like how, which reflects how vascular, how change in cerebral blood flow in a given brain region provoke change in other brain region, right? Or change in cerebral blood flow in another region. And we also consider structural connectors, which are the ones that we estimate from fiber with fiber radiography techniques. And in practice, what we do is that we collect different imaging modalities for each participant. Like in this case, for example, we collected for this study from 2017, we collected arterial spin labeling, which allows to estimate cerebral flow in the brain. We also used amyloid PET, resting state fMRI, from which we obtained indicators of functional integrity, like rigor, amplitude of the low frequency signals, the connectivity between the different areas, or the correlation between different areas, actually. Glucose metabolism PET, the structural imaging or structural MRI, from which that we use it to estimate gray matter density and volumes of the different brain areas that we consider, and also cognitive information. So then we extract the regional values, and we define this state space vectors, in which you can see that what they have is just information on, let's say, the vascular flow for a given set of regions concatenated with the amyloid deposition in the other regions. And then, like, essentially what we have is like a vector with all the values that we extracted across the different imaging modalities for a given time point. Or also we can define it for a given clinical group, right? For the person that are only control, for the person that are at early mild cognitive impairment, or for the person that are with Alzheimer's. And then this allows to then estimate this big matrix, in which what we have is how the different regions and specific biological factors that we consider estimated from the imaging data, how they interact across the disease process, and how those interactions project or change or provoke change on cognitive at the cognitive level. That is what is on the right here. And if I represent this in another way, the model allows us to estimate how the different biological factors interact from in a direct way, not in a correlational way. So normally you will see a lot of studies in this area show that extract conclusions or make conclusions based on correlations or covariance analysis across different imaging modality, or let's say considering the covariance between different brain regions. However, we were focused on estimating association that will be reflecting only direct effect of how, let's say, an alteration or perturbation in the vascular system will affect the clearance of the myophotoproteins in the brain, and how those effects will propagate across years to provoke the clinical symptoms that we see at some point in the patient. We can use, once that you have a model or a causal model capturing direct interactions between different biological factors in the brain. In this case, it's only it's only a macroscopic model. So because we are only considering here macroscopic factors measured with brain imaging. And in the second presentation, I will talk about how we can take this to the molecular level too. Once that we have that kind of model, so we can use them to reply on to investigate how probable or how likely will be the different hypotheses that have been proposed for the disease. So if they, for example, if the prion-like hypothesis say that the disease starts in one region and from there propagates to other regions, causing the other or a chain of effects in other biological factors, so we can try to see if the model replicates that in reality. So we can inject a perturbation, a virtual perturbation in a given brain region, and see how much that provokes the change that the prion-like hypothesis proposed that occurs. But we can do the same associated with to simulate the metabolic, what the metabolic hypothesis proposed, and the same what the vascular hypothesis proposed. So we can, this kind of mathematical modeling, essentially, once that we feed the data, that we identify the parameters of the population, and if they represent well the data, if they reproduce well, or they fit well the data, then we can use them to interrogate and to get closer to biology, which are at the end why we are here, right? And not only that, once that let's say we go back, and if this allows us to understand better how the different biological factors interact, then we can also simulate which would be the effects of therapeutic interventions at the group level or the individual level. And I will talk more about that in the second presentation, but essentially we can simulate how a perturbation in a given brain area and a given specific biological factor, say perturbation in the vascular system of a given brain area, will cause change in the other regions, and we can follow and see if those changes will have a positive or a negative effect at the clinical level, right? So that's close to what the control theory normally does. In this case, for example, for that study, we use the model to integrate back in time. So we feed the data of the 700 subjects, and we integrate it back in time to identify which were the most likely perturbations that conducive to the Alzheimer's data that we had in our hands at that level. And then we observed that a vascular perturbation was a likely event to conduce or to provoke or to reproduce the data of that population, which aligns with the fact that there are many reasons why the vascular system could be an early triggering or could have an early role on the development of Alzheimer's disease. For example, proteins and whey products clearance depends on the vascular and the lymphatic system flows in the brain. And we have described all these mechanisms in a review articles explaining how the vascular system have a major role on the development of Alzheimer's disease and related disorders. What I have presented until here aligns relatively well with this schematic graph proposed by Brown and colleagues in 2017, in which that suggested that to study neurological disorders, which using anatomical and neuroimaging information, we could go from the basic or more empirical models, like just analyzing how the connection change, for example, or using graph theoretical measures, to start integrating those information with or aggregating information across different layers of biological entities, and pass from those more complex, multi-layer, multivariate or multifactorial models to generative brain models, in which we pass from just observing the data or just analyzing associations or difference between groups to try to generate how the brain works in a given context, right? So we can use you probably know or have heard about the neural mass models and how they allow to recreate brain functioning, assuming or after assuming a specific way of interaction between neurons at the regional level, and how those interactions and interaction between region to region neural mass in the whole brain. But we can also do the last model that I presented, the multifactorial causal brain modeling, is an example of generative brain modeling that allows to assume a specific potential ways of interaction between the different factors in a flexible way, estimate the parameters controlling those interactions, and then generate new data to study that disorder. Then they also propose to use network control theory to see how the brain, once that we have generative brain models, that could be an individual or the group level, how we can use those to study what specific signals or input signals will need to be introduced in the brain. And when I say introduce it, we can assume, for example, or we could think, for example, in therapeutic signals, right? to conduce the brain from one given condition to from one pathological condition to a desired healthy or at least a stable condition. And I will show some of our studies doing that that have already taken that to a next step. However, in general, we have taken this further, not only focus on brain networks, on brain connections, in order to following an integrative perspective in which we consider that integrating different layers of molecular information from the very high resolution molecular side, which could be genes, proteins, DNA methylation, signals or profiles, how we can integrate this with multimodal neuroimaging, which could be molecular imaging or structural functional imaging, with lifestyle conditions, clinical information, therapeutic information, in order to create these more integrative networks, not only brain networks, but whole body-brain interactional networks that will allow or could allow us to better understand complex disease mechanisms that can only be explained with one biological factor, and also that could allow us to better estimate or identify treatments at the individual level that could be more effective and conduce the person from a given pathological or move the person from that is following a given disease variant to another trajectory or more healthy trajectory. And we'll, in the next presentation, focus on other types of modeling with other type of data and how other information can be integrated to reply the same questions that we have been trying to reply from the beginning here and other associated questions. We have also worked on sharing these modeling techniques because we know it's challenging, it takes a lot of time, and also it's good to get feedback and to see how they apply or not in other contexts and for other, when studying other neurological disorders. And here, for example, we have I'm illustrating this software that we shared about 2 to 3 years ago that allows to incorporate from genetic, proteomic, any type of molecular information, post-mortem neuropathological evaluation with multimodal neuroimaging data in order to estimate or to apply the models that I just presented and many others that I will present in the next presentation. But essentially, the two main roles of the models that are here are one, first, to clarify disease mechanisms. Again, we can do that focusing on the group level, on the subpopulations, or at the individual level. And second, on identifying the optimal treatment interventions for each individual. We will discuss more about this on the next presentation. Thank you for listening.
Thank you, Yasser, for this very exciting, overview of the research in neurodegenerative disease, in particular in Alzheimer's disease. I think that there are a lot of questions, and okay, yeah. Go ahead.
Yeah. Can you go to the very first slide?
This one.
Yeah. So sincerely, I was not aware. So it's the very first time. So thank you for, and thank you for the great talk. It's very nice. So this is it's shocking, right? So now the very first comment I would do if I see these results is that I could just say, oh, big pharma is very powerful and they can deliver drugs and put in the market drugs that have not been tested properly. That's the very first comment I would have, right? Because to have this kind of failure means that the drug was not tested in the right population at the very first place. Okay, that's the very first comment I would have. And second, so of course, so said that, then I would say, okay, let's test this drug better next time, right? And because you mentioned that, and I think it's becoming clear that we want to do personalized medicine, right? So my fear is that we have tempted to use these results to say, oh, look, we need to go for personalized medicine because just to treat the patient in a like statistical, right? In a statistical way may not help, right? Because look how the medicine are failing, how the treatment are failing. So we need to personalize. So I think that before going to personalization, we have we need to first see if at the group level things are working or not. So that's my very first question. Do you agree with that?
Yeah, well, yeah, this is a complex case, right? So generalized medicine, we know, well, the ideas of personalized medicine are coming not from 50, 60 years ago, they are coming from 2,000 years ago. We just don't know how to implement it. We never knew how to implement it well. So I think generalized medicine is not a total failure. So clinical trials, of course, there are many challenges with clinical trials. As far, I will not enter in how FDA approved drugs or not, because that's another complex thing. But I think that some drugs have been approved as far they prove to improve the condition of a subset of the population with that diagnosis. And then obviously there are others that would be challenged with personalizing medicine, like what does it mean to personalize medicine? Like to have a different drug for each person? How, like, do we need 7 billion of different drugs, or one so specific that it only works for one and not necessarily for the others? We target that questions in some studies, and I will talk a little bit about that in the other presentation. Unfortunately, we find that there are subclusters that could be say representing well the treatment needs without having to go to define a drug for each individual subject. About if we need to study better the groups, yeah, definitely. I mean, one of the issue is not only, let's say, of how we apply generalized medicine and how we will apply personalized. I think that even the drug discovery field needs to revolutionize itself, right? Just to put an example, right now there are two or three drugs approved for Alzheimer's. They were approved in the last 3 years or something. They are targeting very specific subtype of Alzheimer's, right? Which is the more amyloid, the stereotypical amyloid tau Alzheimer's patient. The first one didn't have so clear effects, and then people think that to some extent it was approved due to the urgency to at least have something that will improve some and a small subset of the patients. The second one have a stronger clinical effects, and however, it seems to have a stronger risk too for the population. So just during the clinical trial, I think that 2 or 3 patients died of complications, like they have a global brain hemorrhage. And so it has been, there was a lot of discussion, and there's a lot of discussion if then we need to screen better who take the drugs, because the person died just like after a few hours of taking the medication. That's terrible. So the person goes to participate in clinical trial and dies in the clinical trial. And then what seems to happen that person have vascular complications, which are another comorbidity of these disorders, right? And in the combination of having of trying to remove the amyloid from the brain, in a person that have high vascular damage on the brain, they trigger a chain of alterations or of damage that could not be controlled in time. And in the person that are proposing that drug or that are, let's say, in favor of that drug, what they suggest that, well, then what we need to do in the clinic is to be sure that someone takes the drug don't have the vascular complications. Still there are risks there, right? Because still, how do you control that perfectly? It's so difficult to be sure of that. And so I think that, yes, we need to see, first we need to understand better the disease. So we are really, throwing rocks in the dark here still. So we're not totally sure about what are the causes of almost any neurodegenerative, mild neurodegenerative disorder. Because all the hypotheses, even the stronger ones, fail or are under discussion to some extent. All of them, like the amyloid one, like all of them, literally. So we don't understand at the deep level of the molecular processes what is happening there. So then we are just trying, well, I say we, humanity as a whole, is just trying to, is targeting the major factors that get affected, but without really a causal understanding of how altering these factors will, what will happen after altering these factors. I will show in the next presentation that it's a network what we have, right? If we go to this network here, of network of interaction between different biological factors, and this one is only at the macroscopic level. Let's take it that here. So yeah, so amyloid is here, but because it's a network, and anyone coming from mathematics physics know that, and the physics physics know that, you could always find a signal to inject on any point, on any node that will cause change on the other nodes on the network. If you find or you inject that signal with enough energy, you always will change that. It's like you always can provoke, let's say, an amyloid deposition decrease by targeting functional factors. So I think that the medicine until now have working with the hope that, say, targeting a given set of factors will cause change on this level. And sometimes that works well for some persons, close to it works relatively well. For many others, it doesn't work. So I think that I agree with you that we're not, let's say, I will not try a clinical trial for it, different for each subject. That's also risky. We kind of need to identify subgroup subjects that might have the same needs, and then focus on still understanding, well, when I say same needs, we need to understand why they have those needs, and trying to characterize better, the whole process of developing the drug, or might need the drug, but it could be all about intervention, essentially. I'm not sure if I reply.
Yeah.
Totally, but
I have 2 questions and a half.
Mm.
So the first one is when you show the graph about so people who have ApoE4 have reduced clearance of
yes.
amyloid, is that like a relative value or is that absolute? Because I'm assuming if you have more amyloid, the amount you would clear might be the same as someone else, but relative to what you have would be less. So because I can't
exactly.
see.
Yeah, this is the number of ApoE4 allele copies.
Mm-hmm.
And then the person that have 2 copies, they have significantly less clearing rates according to the
Clearing rate.
Yeah.
Okay.
So it's like for every unit of amyloid deposition, they can clean less and less.
Okay,
Yeah.
got it.
And then we found, so these have been also tested in animal models and with obtaining similar results. And we know that ApoE4 have different implications. It's not totally clear yet all the mechanisms that the ApoE4 can contribute to Alzheimer's disease. Seems to be like associated with lipid metabolism, vascular effects, but other people say that it also contributes to the production of the photoproteins. I think it intervenes at multiple levels.
Okay. And my second question was, is kind of the main factor that dictates the propagation of amyloid between a region and another region, the amount of connections, or is there, so my question essentially is, are some regions more protected or more
Yes.
sensitive, like
Good question. Yeah,
susceptible?
exactly. So there are studies focusing on the regional properties that contribute to increase the vulnerability of that region to accumulate toxic proteins, or to produce or clean less and accumulate not only toxic proteins but also hyperactivity, atrophy, etc. And definitely so. When we look to, let's say, how gene expression changes across the brain, those regions that accumulate or produce more for the proteins have different patterns of gene expression activity than the ones that don't. So that have generated a discussion, and I'm happy that you bring this point. So another possibility is that in a scenario in which the before the proteins of whatever effects, let's say atrophy, doesn't really propagate that much. But because the regions are so similar, so the regions that are connected, they are also similar at the molecular level and the cyclotectonic level, etc. So it could be that the regions are so similar molecularly that they also work in similar ways and they accumulate similar type of proteins, similar kind of dysfunction. And if we only look to imaging data, we might have the impression that, oh yes, there are covariance patterns between the regions, or because they are connected, it means that the information of whatever happens propagates. And there are studies questioning that. And we actually have studied that too. So we have decomposed the second model. This one, Ahmed will be talking about that, and me too in another presentation, in a way that each of these interactions can be decomposed in terms of the local molecular properties of the regions. So we can see at the end, it's like decomposing the whole problem. We can see what component of the propagation, of the increase on a given biological pattern in one region is explained by the local molecular properties. What component is explained by the local interactions, and what other component can be explained by the spreading. So when we decompose this in a competitive way, we have seen that the propagation for some specific biological pathway is not the dominant component.
Okay.
So local interactions, molecular interactions seems to be having a major role too.
Okay, and I guess as a follow-up, then would you also assume that the capacity to clear in different regions would be different, even though they're
absolutely, yeah.
I see.
Yeah.
Oh, that's so interesting.
The capacity to produce, to clear, like to have hyperactivity in a region depends on the local molecular properties. We will enter more in molecular characterization in the second part.
Okay.
But definitely, I think that we cannot remove, let's say, for functional activity, removing the connections. It's not going to work. We know that there's functional signals propagate, the nervous system, etc. For even like the structural alteration, we know the Wallerian degeneration, if we cause damage in one region, structural damage, we will see that spreading, like literally the axons will spread the information structurally, etc. But how the region works internally have a major part too.
Cool, great, thank you.
Yes.
Thank you. Any other question? Okay. Okay. I have a few.
Yes.
But I am going to read it from beta amyloid is thank you. So beta amyloid is a poor predictive factors in Alzheimer's disease. But this is something that you mention, you integrate. So probably the interaction between beta amyloid and other factors can be predictive, but beta amyloid itself. So what is the, okay, what do you think about this first?
Yeah, well, friends are not I would say the major fan of the amyloid cascade hypothesis, but that doesn't really matter here. That's more like personal. In terms of association with structural damage and clinical advances, tau is a better predictor. So tau, however, tau is associated, the appearance of tau is associated, or the spreading of tau is associated with amyloid presence, etc. I think that amyloid is too specific, so it's almost everywhere. If you look to the brain of the person that develops Alzheimer's, you find in so many areas that is not that specific as tau. However, I don't think it's either that tau is the major responsible biological factors of the Alzheimer. I think that there are other, like from early age, there are molecular processes going on that accumulate more or less or have an interaction with environment, lifestyle, genetic factors, etc. And then they start contributing silently. They have been shown that the brain or the brain connectivity of person with say 0.4 is already different at early age, at 30 years old. So, and the vascular system too, before they have any amyloid and tau in the brain. I don't have an amyloid hypothesis to propose right now in terms of biological mechanisms, except to say that I think it's highly multifactorial. I think that for some persons, the causes might be different than for other persons, even when they finish with very similar clinical manifestations. And I think amyloid and tau are like secondary products of things that happen more at the deal molecular level, which is something that we are studying. We are currently studying in collaboration with many other groups. So we show one example here that as illustrating how we try to go back in time, independently of amyloid and tau or atrophy, to identify the timeline, disease timeline of molecular change, and what are the major factors in that disease timeline.
Okay, so regarding the different predictors, you see that the vascular aspect can predict highly the disease. That's what you mentioned.
In this population, yes. I do think that amyloid and tau have a toxic role at some point of the disease. But I also think the vascular deregulation plays a major role in combination with amyloid and tau.
Yeah. So just to mention, what is your opinion about the link that we can observe? Let's say for the beta amyloid, we know that the beta amyloid is really very close to the oligodendrocyte.
Yeah.
And people try to see, okay, why and what is the effect on those kind of cells? And what we see is that the oligodendrocyte produce more glutamate in the presence of beta amyloid. So they release the glutamate in the extracellular space in a more abnormal way. So one effect or one target can be, oh, what's happened with this increase of the glutamate in the extracellular space because of the beta amyloid. In another side, tau, as you mentioned, is something who make the astrocyte not uptaking enough glutamate. So the combined effect of the beta amyloid and tau is this indirect effect of increasing of the glutamate in the extracellular space. This might explain the fact that we have this hyperactivity that you mentioned because of this increase of the glutamate in overall the brain. So, and this probably has also an effect on the vascular aspect. So, and this is the indirect effect that you see. Vascular aspect seems like predictive. So I like having like a kind of hypothesis on those factors. So your model are, I mean, they're correct. They're showing, okay, which parameters seems like predictive. And in general, what is your, because you are moving to this multi-scale approach. Okay, so my question is how we can move to this multi-scale approach without having this kind of hypothesis. I mean, we should have something like at the level of the gene, at the level of the cells, and which cells, or you mentioned the different cells, and now how we can integrate those cells at the level of the population of cells, and then moving to the organ. So what is your opinion on that?
Yeah, definitely. So we are working on that. That's one of the earliest slides that was part of one of the earliest slides that I will present after, in which we have combined epigenetic, proteomics, different levels of biological entities, with the idea of trying to reconstruct trying to reconstruct how alterations in these levels occur during the whole disease timeline. There are some limitations there, so it doesn't exist yet a data that could be good enough to capture the whole disease timeline for neurodegenerative disorders. It is starting to be created in some places now, and I can talk about that too. But we study also how change cell alterations occur with this disease timeline. I didn't show it here, or I don't have it for the next presentation, but we have analyzed it also, certain how the spatial alterations of certain different neurodegenerative disorders associated with the distribution, brain distribution of different cell types. And we have found that a major role for microglia, astrocytes, so the ones that are not necessarily, let's say the more microglia or the less or more, I don't remember now the direction, but you have in brain regions, it associates strongly with the level of atrophy that we will have in Alzheimer's and Parkinson and certain diseases. We suggest that historically, there is something called the neurocentric view of disorders in general, of brain disorders in general, and actually all field neuroscience, even start with neuro, right? So we mostly cared about the neuron for centuries, or almost only cared about neuron for centuries. Well, fortunately, not some people that study all the same types, but when we analyze multicellular type of data, we see that all the cell types have equal important role as the neurons, sometimes even more for some type of specific disorders. So for that, we are analyzing single cell RNA data. We are also cross-analyzing single cell epigenetic data. I will talk at the end too about that. There are very good studies now, or technology now, mapping spatial single cell transcriptomics, which I think will help to reply to some of the questions that you're making. So essentially, these studies are allowing to see what specific genes and what specific cells are getting, active or altered, or are they getting dysregulated at the same time that amyloid, tau, atrophy, tissue damage occurs, right? And another relevant question is not only, because at the end, what we really get worried about, it's not about having amyloid in the brain or tau, right? Or like we don't care about the black box as soon the black box doesn't start malfunctioning, right? What we care is about the clinical, we care about the mechanisms, but at the end, we worry because we have consequences. So damage on the black box have consequences clinically and cognitively. So that's why studying resilience to damage on the black box is so important too. So as like David Bennett from the Grossmark group has said, so at the end, if we find ways to increase that resilience, we might not need to be, like, why do we need to necessarily focus on amyloid, on tau, or removing them? So if we find a way why a person, despite having amyloid or tau, still is functional cognitively, and that's good, could be good enough, right? Or it's an alternative way to keep the person functional.
Okay, so the last question, and then we move to the coffee break, is at this level, so you are not dealing with the black box. I understand this. So, but you introduce these multifactorial models, and multifactorial causal models. What do you mean by causal?
Yeah, causal is that we, in the model, we always try to characterize how properties at a given time induce change in those, say, property of factors at a given time induce change in factors in the other factors in the future. So what we estimate is, let's say, how, say, vascular flow now across brain regions interact with an amyloid to provoke atrophy in the future. So we try to disentangle between direct, statistical direct and indirect effects. And then because we have something on how the past predispose the future, then that's what we mean in this case.
I think the best wording is what you say, direct and indirect effect. Because causality is something like can refer to another concept, which is a little bit more complicated in all our studies. But anyway.
We have, of course, difficulties to prove whatever causal link we estimate. We have difficulty to prove that is for real, right? Because the ideal scenario to prove something is to have interventions. Without, if we are not moving, changing things experimentally, we cannot conclude that there is a cause effect.
Okay, so thank you very much. We are going to stop this first part now, moving to the coffee break, and then see you.